Welcome back to the SANS Science Experience, where we explore the most exciting new research papers and preprints just for you. If you're enjoying these deep dives into the latest science, please do hit that like button and subscribe. It really does help support the channel. Today, we're diving into a really fascinating study. It kind of throws a curveball at how we thought the liver produces energy.
Yeah, it's pretty significant research, challenges some long-held ideas in cell biology.
So we've got this compelling paper looking at the liver's energy production machinery.
That's right. You know, for years, the textbook picture has been pretty clear. Complex 1 is the main way electrons get into the mitochondrial electron transport chain.
Okay, the ETC. That's the cell's power plant, right? Inside the mitochondria.
Exactly. Think of it like a biological circuit. Electrons from food get passed along these protein complexes, and that movement, that electron transfer, it creates this gradient, like a tiny electrical charge difference, which then drives the making of ATP. That's the energy currency cells use for, well, everything.
Right, ATP. Got it. And complex 1 has always been seen as step 1, the crucial entry point, especially in the liver.
Precisely. The primary gatekeeper, so to speak.
But this new research, it looks closely at complex 1 and also complex 4, just in the liver cells of adult mice. And it asks this really basic question. Is complex 1 really essential for the liver? Like, what happens if it's just not there?
That's the core question. And to figure it out, they used some clever genetic tools. They specifically deleted a key part, a subunit called infusion of complex 1, but importantly, only in the liver cells, the hepatocytes of adult mice.
Okay, so they could isolate the effect just in the liver?
Right.
What did they find?
Well, this is where it gets really interesting, and frankly, quite surprising. The mice, they seemed completely fine, no obvious health issues, no signs of liver damage, nothing.
Wait, really? With complex 1 completely knocked out in their livers?
Completely knocked out. And their liver function, totally normal.
Wow. So things like blood glucose, proteins, bilirubin, all the standard checks.
All normal. Fasting blood glucose, albumin, bilirubin, even those liver enzymes, ALT and AST. Everything looked good.
That's kind of hard to believe. What about the actual energy levels, the ATP? Surely that must have dropped.
You'd think so, wouldn't you? But no. ATP levels remained unchanged.
Okay, that really challenges the idea of complex 1 being the main energy gateway.
It absolutely does. And they also look for oxidative stress, you know, that damaging imbalance often linked to mitochondrial problems. And again, not much happening there. The glutathione ratio, other redox markers, mostly normal.
What about those NAD plus NADH ratios? Those are usually quite sensitive to complex 1 issues in, like, cell cultures.
That's another key finding. In these living mouse livers, those crucial NAD plus NADH and NADPH, NADP plus ratios, they weren't altered. It's a stark contrast to many in vitro studies.
So the liver in the actual animal seems to handle it differently. Did they look inside the cells, like cytosol versus mitochondria?
They did. They checked the redox status in those specific compartments too. And again, normal. It suggests the liver has robust ways to maintain balance.
So was the liver compensating somehow? Did it have to, like, ramp up other systems? Did they look at proteins and metabolites?
Good question. They did extensive proteomics and metabolomics looking at thousands of proteins and small molecules. And the results were striking for how little changed. Very few proteins were up or down, mostly just other parts of complex 1 itself, likely because the complex became unstable without infusion.
So not a massive rewiring effort.
Doesn't seem like it.
No.
Only one metabolite, thiamine monophosphate, changed significantly, and they weren't sure why.
Incredible. It's like the liver just shrugged it off. Did they check how it used fuel, like glucose?
Yep, they used labeled glucose tracers. No significant differences found in how the complex 1 deficient livers process glucose or fed it into key pathways.
And oxygen. Since the ETC uses oxygen, did they measure that?
They did, using sophisticated MRI in live mice to measure oxygen levels in the liver tissue. Even when they challenged the mice with high oxygen, the complex 1 deficient livers consumed oxygen normally. Their tissue oxygenation stayed normal.
So the bottom line for complex 1 is, it seems surprisingly dispensable for normal liver function in these adult mice under standard conditions.
That's the main conclusion for complex 1, yes. Quite a departure from the classical view.
Okay. But the study didn't just look at complex 1, they also investigated complex 4, right? As a comparison.
Exactly. And that comparison is really illuminating. They knocked out a key assembly factor for complex 4 called COX-10, again, just in the liver cells.
And what happened there? Was it the same no big deal story?
Oh, not at all. It was a completely different picture, a stark contrast.
Okay, how so? What problems did they see with complex 4 dysfunction?
These mice developed significant liver issues. Their livers got larger, looked pale, signs of fat buildup, which is called steatosis. They also had less stored glucose, less glycogen, and lower fasting blood sugar levels.
Ah, so clear signs of metabolic trouble. What about making new glucose? Gluconeogenesis.
That was impaired too. They showed that using a lactate pyruvate tolerance test, the liver was struggling to produce glucose when needed.
And those standard liver health markers, ALT, AST, albumin.
Those definitely showed problems. Lower circulating proteins like albumin, higher bilirubin, suggesting impaired function, and elevated ALT and AST indicating liver cell damage.
Right, so complex 4 loss definitely causes liver damage, unlike complex 1 loss.
Right.
What about ATP and redox status in this case?
Interestingly, overall ACP levels didn't change dramatically here either, which is perhaps surprising given the dysfunction, but they did see a trend towards more oxidative stress, and critically, the NAD plus NADH ratios were significantly altered.
So a clear disruption in the redox balance.
Yes, both in the cytosol and the mitochondria. Complex 4 dysfunction definitely throws that internal balance off kilter.
Makes sense. So what do the deep dives into proteins and metabolites show for the complex 4 deficient livers?
The proteomics showed a big increase in many other mitochondrial proteins, maybe the cell trying to compensate, but also downregulation of complex 4 components themselves, as you'd expect. Gene analysis pointed to, an increase in mitochondrial stuff overall, but also a downregulation of metabolic pathways, especially fat metabolism.
Which fits with the fatty liver finding and metabolites.
Very significant changes there. An increase in 2-hydroxyglutarate, that's a known marker for mitochondrial problems. Also, higher bile acids consistent with bile flow issues, cholestasis, and perhaps the most telling thing, a big pileup of long-chain acyl-carnitines.
Acyl-carnitines.
Okay.
Those are involved in fatty acid breakdown, right?
Exactly. Their accumulation strongly suggests a bottleneck in fatty acid oxidation. The liver couldn't process fats properly for energy.
Okay. And the MRI oxygen measurements, how did the complex 4 livers handle oxygen?
They struggled. Unlike the complex 1 livers, when these mice were given high oxygen, their liver tissue oxygen levels didn't respond properly. It indicated impaired oxygen consumption.
Which makes sense if complex 4, the final step that uses oxygen, isn't working.
Precisely. It directly impacts the ability to use oxygen.
So putting it together, complex 1 loss, liver seems okay. Complex 4 loss, liver is clearly sick, damaged, metabolically stressed, can't handle oxygen right.
That's the picture. It strongly suggests that while complex 1 might be bypassable, the electron transport chain itself, particularly the later stages like complex 4, is absolutely vital for liver health.
So if electrons aren't primarily going through complex 1 in the liver, where are they coming from? What are these alternative routes?
The researchers propose a major role for something called the electron transfer flavoprotein complex, or ETF.
ETF complex. What does that do?
It collects electrons mainly from the breakdown of fatty acids and also some amino acids. And crucially, it feeds those electrons into the ETC downstream of complex 1 directly into the ubiquinone pool.
So it's like a side entrance that bypasses complex 1.
Kind of, yeah. And the buildup of those acyl-carnitines and that 2-hydroxyglutarate-like metabolite in the complex 4 mice, that pattern is actually very similar to what's seen in humans who have genetic defects in the ETF system.
That strengthens the case for ETF being important here.
It does. Plus, they know the liver generally has pretty low activity of the enzyme PDH, which links glucose breakdown to the TCA cycle, the main source of NADH for complex 1.
Right. If the main on-ramp for complex 1 isn't that busy anyway in the liver, maybe losing complex 1 isn't such a big deal.
That seems to be the implication.
Now, are there caveats? This was in healthy mice, specific strain, standard diet. Could complex 1 be more important under stress or different conditions?
Absolutely. That's a crucial point the authors make too. This was under baseline healthy conditions in C57BL6J mice. That specific strain also has a known genetic quirk affecting how ETC complexes assemble into supercomplexes, which could influence electron flow. So yes, complex 1's role might be very different under metabolic stress, different diets, fasting, or in other genetic backgrounds. More research is needed there.
Okay. And what about the bigger picture? Like, for mitochondrial diseases, they often affect specific tissues differently.
This could offer a really important clue. Maybe different tissues rely more heavily on different electron entry points. It could help explain why complex 1 defects often hit the brain and muscles hardest. Perhaps those tissues rely heavily on complex 1.
While the liver, maybe using ETF more, is sometimes spared.
Potentially. And conversely, complex 4 issues might have broader effects because it's essential regardless of where the electrons initially came from.
So really understanding these tissue-specific fuel sources could be key.
Definitely. It could lead to more targeted therapies for mitochondrial disorders, focusing on the specific pathways active or deficient in affected tissues.
So what does this all mean for you listening? Well, this research really shakes up a fundamental concept in cell biology. It suggests our livers might have a clever workaround for energy production that we didn't fully appreciate. The idea that complex 1 isn't essential is, well, it's pretty groundbreaking.
Yeah, and here's where it gets really mind-bending, It forces us to ask, what exactly are these alternative fuels and pathways feeding the liver's energy system? Are they fatty acids via ETF, amino acids, something else? And how does their importance change depending on, say, what you just ate or if you're fasting?
Which raises a huge question. If we understand these alternative routes better, could we potentially leverage them? Could we support liver function in diseases where complex 1 is a problem, or where energy supply is generally compromised? And could this explain why some mitochondrial diseases seem to skip over the liver while devastating other organs? Lots to think about.
It really is. We definitely encourage you, if you're interested, to delve a bit deeper, look up the electron transport chain, ETF, complex 1, complex 4, and just think about the implications of tissue-specific energy strategies for our overall health.
Absolutely. Well, thank you for joining us for this deep dive on the SANS Science Experience. If you found this interesting, please hit that like button and subscribe for more explorations into the cutting edge of science. We'll catch you next time.